Chapter 15, Carbohydrates of Physiological Significance, presented by Garrett Johnston. I'll start by going over a few key points from this chapter, and then I will explore them more thoroughly in the subsequent slides. Carbohydrates are the primary source of energy for cellular metabolism, especially during moderate to high-intensity exercise, when carbohydrate utilization far exceeds fatty acid utilization. They serve as structural components for both plant and animals. They're involved in cell-to-cell communication as constituents of cell surface molecules, as can be seen in this diagram. They're also implicated with several metabolic diseases, most notably diabetes mellitus, which is a growing public health concern. There are 4 classifications of carbohydrates based on the size, or rather length, of the carbohydrate chain. Monosaccharides are the smallest unit of sugar and are unable to be hydrolyzed into simpler units. Disaccharides are the condensation products of 2 monosaccharides. For example, the condensation product of glucose and fructose, which are both monosaccharides, is the disaccharide sucrose. Oligosaccharides are the condensation products of 3 to 10 monosaccharide units. They're often indigestible and are commonly constituents of cell surface molecules that engage in cell-to-cell communication. Polysaccharides are condensation products of 10 or more monosaccharide units and are primarily concerned with energy storage as well as structural support. There are 3 main representations for monosaccharides. The first is the straight chain form, which is the most unstable and is why we often call it a transient state. However, it can be useful for identifying functional groups as well as viewing isomeric relationships between monosaccharides. For example, here in this straight chain form of glucose, we can see that it is an aldose isomer because it has an aldehyde functional group located on carbon number 1. Next, we have the Hayworth conformation, which is a Paranos or 6-membered ring, and it is also helpful for identifying isomeric relationships, especially epimers, as well as alpha and beta anomers, due to the relative ease of identifying which functional groups and substituents are located above and below the ring structure. The most stable conformation of a monosaccharide is the chair conformation. It is the lowest energy conformation and is perhaps the most realistic representation of a monosaccharide that we would see in a biological organism. The first isomeric relationship is L and D isomerism, which are enantiomers of one another, and is determined by the spatial orientation of the hydroxyl group on the terminal chiral carbon of the monosaccharide carbon chain. So in the case of glucose, we would be looking at carbon number 5. So when the hydroxyl group is on the left side of the carbon chain, it is denoted as the L isomer, so we have L-glucose. And if it's on the right side of the chain, we call it the D isomer, so D-glucose in this case. Alpha and beta anomers are determined by the relative orientation of the hydroxyl group on the anomeric carbon, carbon number 1, to the CH2OH group on carbon number 5. Here's a better example of that on this slide here, which shows that when the glycosidic bond and the CH2OH substituent are trans to each other, we have what is called the alpha anomer, and when they are cis to each other, we have what is called the beta anomer. Furanose and pyranose rings refer to 5 and 6-membered ring conformations of monosaccharides. It's important to note that The majority of glucose molecules found in biological organisms adopt the pyranose or six-membered form, as this conformation has more ideal bond angles and is therefore more stable. Aldose ketose isomerism refers to both the location and type of carbonyl-containing functional group within a monosaccharide. So if we go back up to the structural representation slide, we see in this straight chain form of glucose that we have an aldose, or an aldehyde functional group on carbon number 1. If the carbonyl group was on carbon number 2, we would have a ketone functional group, and in that case it would be referred to as a ketose. Lastly, we have epimers, which have a diastereomeric relationship to one another, as opposed to the L and D isomers, which were enantiomeric. Epimers like L and D isomers are determined by the configuration, or the spatial orientation rather, of the hydroxyl group. However, with epimers, we're only looking at a single carbon atom. So the spatial orientation of hydroxyl groups only differs at a single carbon atom when we're talking about epimers. So for example, when glucose is epimerized at carbon 2, we see that all hydroxyl groups within this pyranose or Hayworth structure are facing in the same direction between the when we compare the glucose and mannose form, except for carbon number 2, where in glucose the hydroxyl group is pointing below the ring and the mannose group is pointing above the ring. Similarly, glucose epimerized at carbon 4 yields galactose, which is epimerized at carbon number 4. So in alpha-D-glucose, the hydroxyl group at carbon number 4 is pointing below the ring, and at carbon number 4 of galactose, it's pointing above the ring. Glycosidic linkages represent the progression of a hemiacetyl to an acetyl, which is characterized by a condensation reaction that results in the loss of water. Nucleophilic attack at the anomeric carbon can generate both alpha and beta glycosides. It's also important that the reverse reaction of condensation, hydrolysis, can also occur, which results in the cleavage of the glycosidic bond back into its simpler monosaccharide units. When we compare glycogen to starch, we see several similarities and differences. They're both storage molecules, storage polysaccharides specifically. However, glycogen is found in animal cells, whereas starch is found in plant cells. They both contain alpha-1,4 and alpha-1,6 glycosidic linkages. However, starch is composed of two glucose polysaccharides, amylopectin and amylose, where glycogen is only composed of one glucose polysaccharide. Glycogen contains more extensive branching than amylopectin, and amylose is completely unbranched. Branching expedites or mobilizes energy during glycogenolysis, which is particularly advantageous for animal cells during periods of high energy utilization. There are several structural carbohydrates, such as GAGs or glycosaminoglycans, which are complex polysaccharide chains containing amino sugars and uronic acids. When they coil around a protein core, they form proteoglycans. Proteoglycans are a component of connective tissue, and often, because of their ability to absorb water, lubricate connective tissue in a way that facilitates smooth and pain-free movement. Chitin forms the exoskeleton of crustaceans and insects and consists of the N-acetylglucosamine subunit, which contains an amide functional group on carbon number 2. Cellulose is a chief constituent of plant cells and is indigestible and therefore serves as a significant component of dietary fiber. So oligosaccharides form glycosidic linkages with proteins and lipids to form glycoproteins and glycolipids respectively, which are membrane proteins that engage in cell-to-cell communication, recognition, as well as a host of other functions. So an example from the textbook is a transmembrane carbohydrate found in red blood cells called glycophorin. And its roles include serving as a binding site for pathogens and influencing intracellular protein folding and transport. Additionally, due to the net negative charge of carbohydrate chain, they prevent cellular cohesion, which, as you can imagine, is particularly useful for red blood cells where where smooth laminar blood flow is of utmost importance, essentially preventing red blood cells from clumping together. Comprehension 1 says, you accidentally step on the exoskeleton of a cicada during your morning walk. Which polysaccharide is a primary contributor to this crunchy shell? Well, we talked about that glycogen and amylose are both storage polysaccharides, and that cellulose is primarily found in plant cell walls. Therefore, the best answer would be chitin. Comprehension question 2 says, structurally, glycogen contains more extensive branching than starch. What physiological advantage does additional branching confer? Well, the answer to this question is A. Animals can mobilize energy much more rapidly than plants. And the reason for this is that animals often require much more rapid bursts of energy than plants do, whereas plants can sustain, plants don't require immediate bursts of energy in the same way that animals do. Question 3 says, which class of carbohydrates is commonly found attached to cell membranes and encodes biological information? The answer to this would be oligosaccharides, which consist of 3 to 10 monosaccharides and form glycosidic linkages with proteins and lipids on cell membranes, engaging in cell-to-cell communication as previously discussed. Question 4 says, which type of covalent bond links monosaccharides to secondary molecules? Well, as we learned about in the beginning of this class, peptide bonds link peptides or amino acids together to form peptides. Hydrophobic bonds are non-covalent bonds, and they promote the folding of secondary polypeptides into their tertiary structure. Next, we have ester bonds, which are primarily found between glycerol and fatty acids. So the best answer for this question would be B, glycosidic bonds. Comprehension question 5 says monosaccharides can exist as isomers that differ in the location and type of their carbonyl-containing functional group. Which two functional groups are involved in this relationship? And the answer here would be A, ketones and aldehydes. As we discussed, as one of the isomeric relationships are aldose-ketose isomers, which differ in the location as well as type of carbonyl-containing functional group. Thank you.